My name is Lidia Ripoll, and I'm a final year PhD student at the University of Cambridge. I work with Professor William Schaefer and Professor Petra Vertes in trying to understand the brain from a complex systems perspective. Have you ever wondered how does the brain control how you behave? The brain is one of the most complex systems known to humankind. We often imagine it as a giant circuit board with neurons connected with tiny wires called synapses. But what if I told you that there is a whole other way that brain cells can communicate? One that doesn't rely on physical contact between them. Neurons have a group of small molecules called neuropeptides that, like Wi-Fi, allow the communication between two distant points without the need of cables, or in the case of the nervous system, axons. This wireless system is crucial for mood, sleep, appetite, and even some psychiatric conditions like depression and Alzheimer's. All these wireless connections intertwine with the wired synaptic connections, forming a complex system of neuronal communication. However, we still don't fully understand how this network is structured and how it works. In my research, I use network science tools to represent all these connections as a network. Network science is a discipline that represents complex systems as networks. In this case, every node in the network would be a neuron, and every edge in the network would be represented by a connection between two neurons. To build a wireless neuropeptide network, I chose a small organism. The tiny worm can interpret this elegance. Then, combining three different biological data sets, I could identify the nodes and the edges of the network. The nodes are the neurons that send and receive neuropeptides, whilst the edges, in this case, will be given by information about which neurons receive which neuropeptides. Finally, with information about how far are neurons from each other in the nervous system of C. elegans, I could build the first complete neuropeptide network for any brain. One of the advantages of network science is that we can not only represent complex systems as networks, but we can also identify emergent properties and features from these systems in the network. For example, here, in the World Wide Web network, we can see that there are certain instances of certain cities being hubs or highly connected nodes in the network. These are properties that we can extrapolate to any network. For example, in the neuropeptide network, we can look for hubs by calculating the degree distribution of the network. And by that we mean counting all the edges that reach a node in the network. Once we have calculated that, we can identify the nodes that have the highest amount of edges, so the highest degree. Those will be our hubs. If we have selected our hubs, we can also look for another network property that is called Rich Club. And this property identifies whether the highest connected hubs connect more between themselves than to the rest of the network. We can look for hubs on a Rich Club in the neuropeptide network. In order to do that, we calculate the synaptic degree and the neuropeptide degree for each neuron in the nervous system of C. elegans and correlate them, which is what is shown in this plot. Every dot shown in the plot is an individual neuron of the nervous system of C. elegans, and the neurons colored in red represent the Rich Club, which is more than 2/3 of the overall nervous system. We can also see that there's a positive correlation, given that neurons that have a high amount of synaptic connections also have a high amount of neuropeptide connections. Now that we have identified that the network has a Rich Club, we want to understand better how the information flows through this coordinating center of the network. In order to do that, we use dimensionality reduction, a set of techniques that allow us to group nodes based on the pattern of connectivity. In this plot shown here, you can see that each node is a neuron, and they are grouped based on the similarity of their pattern of connectivity. Each color represents a different group. In this method, we find 4 groups. Given the broad role of neuropeptides in health and disease, understanding this network could transform how we approach neurological and psychiatric diseases and their treatment in the future.